Well, thank you, Liz and Wendy, for inviting me to be a part of this just terrific panel of speakers. And I want to thank all of you for hanging in here to the end of a very long day. I have to tell you that when I was an assistant professor, Kafui, I was giving a talk at one of the academic pediatric meetings, and I was scheduled to do the last talk on the last day. Not a great place to be, but I came and I was full of energy, full of enthusiasm for what I was going to do. And I walked in the room and there were two people there. And one of them was the guy who was going to introduce me. And he left after he introduced me. So I'm very grateful. I'm aware that I'm all that stands between you and dinner, and I promise that I will take that responsibility seriously. So my title is Stress in Children, and what I've come to think of as the biology of misfortune. This story begins for me with this iconic figure, this relationship between socioeconomic status on the one hand and chronic disease incidence on the other. This shows in childhood the number of chronic limiting conditions, ear disease, physical inactivity, and so on, as a function of socioeconomic status going from low to high. And it shows this inverse monotonic association that is so powerful that we don't accept any other association without first controlling for socioeconomic status. And yet it is the case that up until about 15 to 20 years ago, we never studied social class as a phenomenon in itself with respect to health. But it is true that poor children sustain higher rates of acute, chronic, and disabling illnesses. It's also true that poor children have higher cumulative mortality and morbidity from chronic disease throughout adulthood. So this shows coronary heart disease as one exemplar, cumulative morbidity over years of time, going from 35 to 75 years of age, and shows those who were brought up in low socioeconomic circumstances within childhood versus those who were brought up in higher, more advantaged circumstances. And you can see this progressive widening of the gap between those who in childhood experienced communities of great advantage versus disadvantage. Now, it is also the case that the conventional mediators of risk that we think about with respect to coronary heart disease or other chronic diseases, things like smoking, cholesterol, obesity, blood pressure, and so on, fail to account fully for this association between SES and health, which leads then to the perennial question of what then are these mediators of social disparities in health. And I want to suggest to you that one of the most important, is the socioeconomic partitioning of stressors and adversity, the way in which stress and adversity is socially layered within populations of children. If we look at children brought up in poverty versus those being reared in middle-income families, and we look at a variety of these kinds of stressors and adversity, density in housing, other housing problems, noise in the ambient environment, family turmoil and chaos, and violence. In this beautiful work of Gary Evans at Cornell, you can see that the children who are reared in poverty have far more of these kinds of adversities and stressors than their counterparts within middle-income families. And it turns out that when we look at all of the details of the differences between children reared in advantaged circumstances versus those in low SES circumstances, there are just these pervasive differences in the experiences of everyday life. They are perennially gnawing, they go on and are part of the mundane character of life in these two kinds of communities. But it is also true, and I think equally interesting, that there is a tremendous amount of variability in the developmental outcomes that we find by adversity and stress. So when we look at the actual data points, these are data from one of my early studies looking at behavior problem severities as a function of family stressors, you can see that there's a tremendous amount of variability in this association. To be sure, a positive, monotonic, and significant association, but there is a tremendous amount of variability here as well. We tend to refer to these children as those who are vulnerable, those who have low levels of family stress, but high levels of whatever the maladaptive outcome is that we're looking at. And we think of these children as those who are resilient, the ones who have high levels of family stress, but maintain relatively low levels of behavior problems or other maladaptive outcomes. It's also true, this is Canadian data, that you can, if you look at the data points for reading scores and literacy levels as a function of socioeconomic status in Canadian children, there is still this tremendous variability that we see in all of these associations with social class. We hypothesized and began to claim several years ago that this variability may be better explained by individual differences in sensitivity to both positive and negative environments. And let me show you some of the data by which we made that claim. So we began questioning whether the noise that we found in all of the variability in these social class health associations was really noise, or whether it was in fact the music that we ought to be listening to. So we began to bring children into very scripted, standardized circumstances in the laboratory, and looking at the two principal stress response systems in the human central nervous system. Those being the corticotrophin releasing hormone, responsible for the output of cortisol, and the locus ceruleus norepinephrine system, which through dopaminergic projections into the hypothalamus is responsible for igniting the whole autonomic nervous system response that we know as the fight or flight reaction. We then began to study these individual differences in biological reactivity to psychological challenge. In measuring autonomic reactivity, we used impedance cardiography. In adrenocortical reactivity, we used changes in salivary cortisol. And we found within all of these samples of children that we examined these very broad and reliable individual variabilities in magnitude and patterns of response. So we had these very standardized scripted measures within laboratory circumstances. We found a great deal of variability. We knew where children were on that spectrum, and we could then take those standardized measures into epidemiologic studies, looking at the actual stressors that children experienced in their day-to-day, week-to-week lives. And here's what we find. This is going to be a cartoon of a recurrent finding that we have had over the last 15 years in looking at pre-syndromal behavior problems, both externalizing and internalizing, in looking at the severity and incidence of respiratory illnesses, injury incidents. We did one study looking at violent injuries in rhesus macaques, and also memory for stressful events. We divided children into those who were being reared primarily in supportive versus stressful social conditions, and we further divided them into those who in the laboratory had shown low reactivity versus high reactivity on this scale of autonomic and adrenocortical reactivity. And here's what we have found fairly consistently over the last decade and a half. When we look at the low reactivity children, we basically find that they are relatively indifferent to the effects of sort of species-typical variation in the supportiveness or stressfulness of their social context. By comparison to that, the high reactivity children show the very highest levels of these maladaptive outcomes under conditions of naturally occurring stress and adversity. We had predicted this part. What we had not predicted and were repeatedly surprised by was the fact that these children who have this risk factor of high autonomic and adrenocortical stress reactivity, when they were being reared in supportive, nurturant conditions in their families, communities, and schools, had, instead of just normative levels of these outcomes, had the lowest levels of these outcomes of all the children in our samples. So these high reactive children had either the worst or the best of the outcomes, depending upon the social context in which they were being reared. We've used this lovely Swedish idiomatic expression, maskrosbarn, which means dandelion child, to refer to the children who are relatively low in reactivity and who show this relative indifference to the effects of supportive versus stressful social contexts. And we coined this Swedish neologism, or orkidebarn, or orchid child, to refer to these children who, like the orchid, have this exquisite sensitivity to the character of their social environment, but when raised in really highly nurturant and supportive conditions, have these exquisitely wonderful outcomes. So we have talked about this as differential sensitivity to social context, referring to these children who have this exquisite sensitivity to the character of the social world. So at this level of the CRH system and the locus coeruleus norepinephrine system, we find this kind of crossover interaction. Now it turns out that almost concurrently with this, Jay Belsky in his work on temperament and behavior began to find the same kind of relationship. So Jay, in looking at differential susceptibility to rearing experience in the case of childcare, has found that those children with high temperamental negativity had either the highest or the lowest teacher-reported behavior problems in kindergarten, contingent upon childcare quality. And that children with low negativity in their temperament were relatively unaffected by childcare quality. So again, this kind of contrast between the dandelion children who show relative indifference to their social environment versus the orchid children who show these dramatic differences in outcome. We've also found the same kind of thing at the level of allelic variation within genes. So Nikki Bush, one of my former postdoctoral fellows and now an assistant professor of psychiatry at UCSF, has studied the BDNF MET carrier, the so-called risk allele, versus BDNF VAL-VAL homozygous children in predicting the level of chronic cortisol secretion in a group of children that varied highly by family socioeconomic status, family income. And she shows that this BDNF MET, the so-called risk allele, the carriers of that allele show either the highest or the lowest cortisol expression, depending upon family income. Again, the orchid and the dandelion. This is strikingly similar to Stefano Guerra's and Fernando Martinez studies of early exposures to microbial products that confer protection against atopic disease. This is the predicted probability of sensitization as a function of endotoxin load exposure, and they study this in the CD14 receptor allelic variant and find this interaction between that allelic variation and the endotoxin load. The T allele diminishes or amplifies sensitization depending upon the endotoxin load to which the child has been exposed in early life. Again, the orchid and the dandelion. So here we now have at the level of temperament, CRH locus coeruleus systems, the periphery, and gene by environment interaction, these same kinds of interactions that we find being replicated at different levels of scale and complexity. So how would we explain this? These are G by E interactions without really an account of how they happen. We have a situation not unlike this famous Einstein cartoon, altered a little bit for the purposes of this talk. We have genes and environments. We find statistical interactions between these two predictors of health outcomes, but it's as if there's sort of a miracle that occurs and we get these G by E interactions. How do these actually happen? Well, we think that they happen by virtue of these chromatin modifications by epigenetic marks, the same way in which these two butterflies, which look like two different species of butterflies, are in fact the same species who have emerged at different periods of time during the seasonal year, and who have epigenetically modified their morphology based on light of day and the length of light over a diurnal cycle. So in order to do an effective job at explaining how this all works, I need to invite you briefly to a reprise of Molecular Biology 101A. And let me just remind you that there is within every gene a coding sequence. There is a start and a stop sequence as well that tells when to start and stop transcription. There is this transcription complex that assembles consisting of transcription factors and RNA polymerase, the enzyme that actually reads the coding sequence. The coding sequence is then read in a process called transcription into messenger RNA. Messenger RNA is then translated into a protein, and that protein is the operative endpoint that then determines the phenotype that is the end product of these complex genetic transactions. But it's also true that there can be risk alleles, as we've just been talking about, let's say in gene X. And when this happens, there is a flaw or a change in the messenger RNA. There is a consequent change in the character of the protein, and there can be a risk for a maladaptive phenotype. But this risk allele, which produces this changed protein, is usually insufficient to produce a maladaptive phenotype. Usually there needs to be some other element, and that is having to do with the promoter sequence that lies anterior to the start sequence of the coding. This is like a dimmer switch which turns on or off the expression of this gene. Again, the transcription complex comes together, and under good circumstances in regard to this gene X, there is a methyl tag that is added to this promoter region. This physical-chemically disallows the assembly of the transcription complex, and then there is of course, a denial of transcription and translation, and then the expression of an adaptive phenotype. On the other hand, when there are traumatic kinds of circumstances that the child may be dealing with, this methyl group can be dismantled. The promoter region is then free to accelerate the expression of this gene. The RNA polymerase transcribes, then translates into the protein, and there results this maladaptive phenotype. So the phenotype occurs only with the combination of these risk alleles from gene X, this hypothetical gene, and environmental adversity. And one possible mechanism of this is epigenetic. It's these marks that are placed in the promoter region of a gene. So we have had a chance to, in recent years, study this phenomenon in the Wisconsin Study of Families and Work with Marilyn Essex. This is a study that enrolled 570 women in the second trimester of pregnancy. They were carefully followed through the succeeding years during infancy and preschool. They reported, both the mother and the father in the couple reported depression symptoms, expressed anger, parenting stress, role overload, financial stress, and then these were looked at as prospective indicators of outcomes down the line. 15 years later, we've had the opportunity in a subgroup of these children to do epigenetic profiling of buccal epithelial cells using the Illumina microarray platform, which interrogates 28,000 CpG sites in 14,000 gene promoters. And here's what we find. We find first that there is differential methylation of multiple CpG sites by parental stress in infancy and preschool. This shows the level of hypermethylated regions, CpG sites within promoter regions. Below the line are the hypomethylated differentially by parents' reports of stressors. The mother's stressors are in red, the father's stressors are in blue. We find that the mother's stressors in infancy primarily are related to differences in methylation for both boys and girls, and we found that the father's stressors in preschool were associated with demethylation differences and primarily for girls. All of these associations were substantial in magnitude, with rows in the range of -60 to +60, corrected for false discovery rates. So what it turns out, it looks as though we are seeing a convergence in this science of gene-environment interplay, in which G by E interactions appear to be mediated by epigenetic changes, which then give rise to these differences in developmental phenotypes. And we now have one lovely example of this kind of mediation of G by E interactions by epigenetic marks. The FKBP5 gene has 4 SNPs. It produces an immunophilin that operates as a chaperone protein to glucocorticoid receptor. Here's glucocorticoid being transported across a cell membrane, being bound by glucocorticoid receptor, and this binding is influenced by the level of expression of this FKBP5 protein. It alters GR function by decreasing binding and impeding the translocation of the receptor complex to the nucleus. The risk allele in this case is a guanine to adenine substitution, and it interacts with childhood trauma to increase the risk of PTSD. The SNPs in the FKBP5 gene produce this increased expression through promoter demethylation, which results in diminished glucocorticoid receptor and HPA dysregulation. So back to our cartoon of this molecular biology. Here are the risk alleles of the FKBP5 gene. Under conditions of trauma, this promoter region is demethylated, which allows the transcription complex to assemble, allows for the decoding of the coding sequence of the FKBP5 gene, gives rise to glucocorticoid resistance and HPA dysregulation, and results finally in adult PTSD. Again, the allelic variation in the FKBP5 gene, along with the early childhood trauma, predicts this outcome of adult PTSD. So here is a G by E interaction mediated by an epigenetic modification. I just want to show you briefly what this interaction looks like. It's an interaction between early trauma in the form of sexual or physical abuse, or sexual and physical abuse. This is the FKBP5 protective genotype. This is the risk genotype. Here is the interaction, the differences in slope, in exposure, and consistent with the differential susceptibility hypothesis, the risk allele is protective in the absence of trauma, but creates greater risk of PTSD in the presence of trauma. So we now have, at these varying levels of scope and scale and complexity, these same kinds of findings of differential susceptibility at all of these multiple levels. I'm going to end with this brief historic interlude. The 1889 German Anatomical Society was the first place where Santiago Ramon y Cajal, the great Spanish neuroanatomist, using the stain that had been developed by the Italian Nobel laureate Camillo Golgi, produced his first pictures of individual neurons and proved for the first time that the brain was not a gelatinous syncytium, but rather was made up of cellular elements. And even more important, showed that there were these points of connection between these various neurons. This is, of course, the discovery that 100 years later has given rise to our whole understanding of neural circuitry, of the communication between neurons, and really provides us with a picture of the physical nexus of interneuronal communication and interplay. I want to suggest that what we are witnessing now in this emerging science of G by E interaction and epigenetics is the possibility of a physical nexus between gene-environment interplay in these epigenetic marks that are placed on DNA and histone proteins. So conclusions: poor children sustain disproportionate burdens of adversity, stress, and disease. Even among poor children, there are important individual differences in susceptibility to adversity. At these multiple biological levels, ranging from temperament to individual epigenomes, reactive children show differential susceptibility to negative and positive social contexts. G by E interactions appear to be mediated, at least in part, by these epigenetic modifications of chromatin structure. and these epigenetic marks may thus constitute a physical nexus of gene-environment interplay. Thank you.